Hello everyone, my name is Thomas. I'm a PhD in plant science in the University Federal of Paraná in Brazil. And today I'm going to show you the SDA protocol or scientific data analysis. This protocol was created by me and Professor Fuxing from University of California, Davis. And before I start, I would like to invite you guys to access this QR code right here. I think it's right here. So this QR code is going to get you redirected directly to this website right here, and you can play by yourself with the options that we have here. So I think it's going to get more interesting for us, and you can access by yourself on the website. So just feel free to access this and we go together. Okay, let's go. Let's start it. So the first step here is just we are going to select some data sets. This data set is called iris. Is basically just for measurements of a flower from the genus Iris. So basically we have Setosa, versicolor, and Virginica, and we have some numerical measurements, okay? So you can select your data set here, and you can select your response variable. So we know that species is our response variable. Also, you can select penguin, is another data set you can play with. Penguin, we have also species as the response variable. But just for today, I'm going to show first the iris data set, okay? So the first step for SDA is to transform everything that is numerical into classes, okay? How are you gonna do that? Basically, we are just using a simple, really simple thing called hierarchical clustering. So I bet you already heard about that. I have like a picture right here. So basically, this is the tree that we have. So this is for sepal length or petal length. Petal length is a good example, so you can see what is going on and how we are going to transform this into classes, okay? So this is just a hierarchical tree, okay? So you can see the true species is right here. And then our goal here is to select a number of categories that we want to transform this petal length. Just by looking, I can see that we can have like basically 3 classes, and that's good enough because first class right here matches perfectly with the species red, which I know is Setosa. Another one here, like a second cluster here, matches perfectly with the green one, and another one matches perfectly with the blue one. So if you have like a small, medium, and high, you have 3 good classes. So intuitively, this is what we are going to do. How we are going to select the number of classes, I'm going to show you in the next step. But keep in mind, this is the goal of SDA, okay? So the first clustering option that we have, the first step, is to select the number of covariate interaction. So basically, if you want to have a hierarchical tree for just one covariate at a time, or if you want to have a hierarchical tree for combinations of covariates. First, I'm gonna run this, and it's gonna be easier to explain you guys every single option that we have once you get the result. Basically, we can control the number of classes that we want here with those options. So first, we have the covariates, we have the clusters, which is the categories that we split. We have the overlapping area, which means that how significant is that category. Here we have the entropy, which is a measurement of association between that category and the response variable. So when you have like a 0 entropy, you means a perfect association. When you have like 1 as entropy, we have like no association at all. First, number of loops. Number of loops is just a way of check if that entropy that you just calculated, like this one here, if this is significant or not. So you're comparing your entropy with random 300 entropies. You see? Okay, so max tree depth, maximum overlapping area, and minimum entropy to stop is a way to control the number of classes that you want. Here you are controlling how you are going to split your tree, okay? So for example here, if I increase the number of max tree depth, I can select it where I want to stop. So if I want to stop like in the first tree depth, just splitting the tree in two, or splitting the tree in three parts, and so on. So maximum overlapping area to stop is basically the algorithm is splitting and calculating the overlapping area from that covariate. So if we start to getting not significant anymore, or the overlapping area starts to increase, for that category, I'm not splitting anymore. I'm just stopping because we are getting not significant values. Minimum entropy to stop is a way to avoid you to create too many covariates. So we have zero entropy here, so this category is already good enough. We have here something around 0.25 entropy, and here as well. So I don't have to split this anymore. We have like, for example, 5 classes, but I could just have 3, you see? And this is actually what happened. So we have just 3 classes right here. Cool. So we created our clusters. Now we are going to go every single iris, checking if the iris has that characteristic or not. So first, we are going to select the characteristics here that we want to use. Okay? So you see we have 30 characteristics here. So we can select the ones that are significant. For example, like this one here. I don't want to have these characteristics because it's not good for me. These characteristics doesn't tell me anything. First, because the entropy is super high, and second, because it's not significant. Okay? This is the third step. We are basically selecting those characteristics. So let's say we have a threshold of overlapping area of 0.1. So everything above this number is not significant for me. So I just want to keep the significant covariates, okay? And I will run the binary matrix. So you can see here that we are going to go through every single iris. So just let's start with this first iris so you have an idea what we are doing here. We have a bunch of zeros and ones. So we are just mapping the presences and absence of those characteristics. The first observation is a setosa. This setosa doesn't have a small sepal length. This setosa right here has a super small C1 is the first class. So super small petal length, and so on. And we have this for every single covariate. Now we have the coolest part here. So we can actually see those characteristics. We can just run a heat map with the presence and the absences. So like petal length C1. So I want to see if those categories has a petal length small. You see a petal width small, and so on. And here in the rows, we have the classes, the true classes like setosa, versicolor, virginica. So we have all the observations, okay? So basically, with the data mechanics, we can just improve the pattern here. Basically, if we increase for 5, now we have 1, 2, 3, 4, 5. So you see, when we increase the number here, it's those are updated. The columns and the rows are updated. So you can see the iris, they were grouped together really nicely. So, and we have those blocks of information with significant information, with information that is important for us. Like we can tell from this map here that this setosa right here has a small petal length and a small petal width. So if you have a setosa, you know the setosa is going to be like that, okay? You have some information right here as well, but this is mostly defines what is setosa for us. And if you have like a petal length a big petal length, you have basically virginica. And you can even see like some kind of subpopulations, I could say like that, because for those right here, the small part, they have some characteristics that the other virginicas doesn't have. You see, like this is really cool because we can map and we can see the characteristics from our data set, and you can control that. But we can go even beyond. So this SDA project here is under development, but we can have really cool stuff with this. First one, I can see like we can have the really good predictions. So if you have like a new iris, you can easily tell which kind of iris is that based on this heat map. So if you have a new iris and you have no idea which species it is, you can ask questions like, is that new iris has a small petal length, a small petal width, or it has like a big petal length, and so on. And with those questions, you can easily decide if that is a Setosa, Virginica, and versicolor. So another cool thing is that you can integrate those informations from the binary matrix with, let's say, ChatGPT or something like that. So you can have like all the information described to you, like someone telling you what is the main difference from those species. So we can have a lot of cool stuff with this. Okay, so let's go again to the original data set. Let's choose like penguins. And for this one, I will do something really different. I'm just like removing one class, okay? So basically, I don't want to see the unique characteristics. I want to see the differences between two classes. So let's start with that. First, we are going to have the clustering options here. I will use like a two, the interaction. So you will see what is going on here. I will decrease this so we will allow to create more classes. Let's see what we get. Oh, see, we have first some covariates that are already categoricals. So we have the island, Bisco, we have island dream, and so on. We can see like really cool stuff like this. We can see like really cool stuff with this. First, the island, we see the island. Those two islands are mostly for one single species because it's zero entropy. So if you have like our island from Torgensen, we have like 47 penguins that are from the same species, okay? And you can see like sex, female or male, it doesn't matter. You cannot classify the penguins using this because the entropy is almost 1, and this is very intuitive, right? Now I select for two-way interaction. So we have like build length with build depth, and it created just 5 classes for that, okay? And so on. This is pretty much the same thing as you saw before. Then we can create our binary matrix. So again, we are just mapping the characteristics from them. Then we can go to data mechanics and run this data mechanics. And look at this. This is really, really cool. So basically, we can see the unique characteristics from those two classes. So let's say, let me just improve this pattern here. So yeah, you see, I think this is good enough. Look how cool this is. So we have those block of informations here, and you can tell, first thing that you can see is that we have a almost perfect separation from the species Adelier from Gen 2. So we can see the difference between those two classes really, really well. You can describe them. So for example here, we know that if a penguin has a build length and a build depth really small, you have Adelier, right? So this is really, really cool. And this is the idea from SDA. You can map the information from your data set and you can see what you have your classes. So you have a completely transparent box here. So this is really, really cool. Okay, so finally, thank you for watching me. Here you have my information. You can just scan this QR code right here and you can have like my LinkedIn. You can feel free to connect with me. You can send me an email and with questions and things, that insights that you had. Also, I have Professor Fushing email here. You can email him or email me. Feel free to talk to us. And that's it. Thank you so much for watching me, and bye-bye.